Hello, my dear students, welcome back to biology classes. So, we were discussing the chapter Biotechnology Principles and Processes. In the last class, we have discussed the construction of the first rDNA. Detail, we have discussed how is rDNA, recombinant DNA produced. The recombinant DNA technology can be called as the modern biotechnology genetic engineering process. So, Stanley Cohen and Herbert Boyer, they have constructed the first rDNA, which was discussed in the last class, and also we have started what are the major tools in rDNA technology. Okay, and in that topic, the tools of rDNA technology, the tools of genetic engineering, we have just started restriction enzymes as the major tool for cutting DNA. Okay, so the restriction enzyme was our topic, and we also already discussed the topic, the first restriction enzyme, and how was the naming of restriction enzymes done, we have discussed. Okay, and also we have discussed what is a palindromic DNA sequence. Okay, do you remember all these things? Okay, so in a palindromic DNA sequence only the restriction enzymes act. You know that. Okay, in order to get the ideas clear, in order to get this concept clear, I am giving a picture representation of EcoR1, action of EcoR1. Okay, in last class you have studied what is the palindromic sequence of EcoR1. Do you remember that? Tell me where the EcoR1 enzyme can cut in a DNA. In a long DNA, which are the portions or wherever the EcoR1 enzyme can cut the DNA? It is a restriction endonuclease enzyme. So, it can cut the DNA in between, not at the ends. Okay. So, an endonuclease, exonuclease difference we have already discussed. So, understand a restriction endonuclease enzyme EcoR1, with a typical example EcoR1, we are discussing the action of restriction endonuclease enzymes. Okay, here I am giving the case of the enzyme EcoR1. Okay, so typical example EcoR1 act, you have studied in a palindrome GAATTC, direction also 5' to 3', this is all you know. This is a simple representation. Okay. Then here the vector DNA and foreign DNA we are introducing, drawing here. I will make it clear to you. GAATTC. Just listen to here. See, suppose this is the DNA segment which we are going to insert to the vector. Okay, in the last class you have studied most commonly used vector is a plasmid. Okay, suppose this is a vector DNA. Okay, and suppose this is our DNA. The DNA which we are going to introduce. Okay, then the plasmid we need to cut first. Okay, then only we can insert our DNA. The plasmid DNA we have to cut, and also the foreign DNA we have to cut and take that particular segment and then to be inserted here. So understand here, this is the foreign DNA. Okay, this is the foreign DNA. Anyone you can take it as a foreign DNA. Okay, whereas this is a portion of the, this is plasmid DNA. Okay. Otherwise, you can just consider this as a vector DNA, any vector. Not only plasmid, nowadays there are many types of vectors. Okay, suppose this is a vector DNA and this is the foreign DNA. Okay, so this is the vector DNA, this is the foreign DNA. For a better ligation, joining, we have to cut both the DNAs, this plasmid DNA and the foreign DNA, both the DNAs with the same enzyme. Okay. Then gluing will be easy. The ligation, joining will be easy. So here we are discussing this foreign DNA we are cutting using EcoR1 enzyme, And the plasmid DNA, the vector DNA also we are cutting with EcoR1 enzyme. Okay, suppose this is a foreign DNA. If we are using EcoR1 enzyme to cut this, we are using EcoR1 enzyme and cutting this vector DNA wherever it cuts. See, it makes cut here in between G and A. So, EcoR1 enzyme cut the DNA between G and A. Okay, here also between G and A. Similarly, if we use the same enzyme to cut the plasmid DNA, vector DNA also. Okay, this is the vector DNA. See, it makes cut in between. It can cut only between guanine and adenosine. I mean, guanosine and adenosine nucleotide. In between guanine and adenine only it cut. So, here it makes cut here. On the opposite strand it makes cut here. Similarly, for the plasmid DNA. Okay, when such a cut is made, it can split the DNA like this. Listen carefully here. When this is cut, this bond is cut, the hydrogen bonds very easily cleaved, and this is the cutting. This way the EcoR1 enzyme cut the DNA so that this portion will be left. Okay, this portion can be released. Okay, like this. That is what is next we are going to draw in the second session. I mean, cut is made like this and that portion is released. Okay, anyway, here I am representing both. So, this is the first diagram where how this is made cut. We are making it clear. Okay, the cut is in between G and A so that the next piece will be like this. Okay, that portion is removed. This much portion is removed. Is it okay for you? So we need this section. Okay, this is cut like this. Okay, so this particular portion is left, the other portion is removed. The next case, similar cut is made there. Then what will be the result? See, the G is cut and removed. Okay, this is the way it removed. And here what is left will be AA, TT, C, and in the other strand G will be left there. Is it okay? Is it clear? Then what happens? This has to be joined. Joining is not done by EcoR1 enzyme. There is the enzyme named DNA ligase. Okay, a DNA ligase action will be easy when both the DNAs are cut. Our source DNA and our vector DNA are cut with the same enzyme. It produces easy sticky ends. These ends can be called as, it's very easily sticking ends. This is called as sticky ends. Okay, this type of cut is called staggered cut. Okay, not a smooth cut, staggered cut, like this cut. Okay, is called staggered cut. And opposite, if the cut is between G and A, if it's okay like this cut means, I mean if it's producing blunt end, not like staggered end, okay, then that is known as blunt end cut. The staggered end cut is shown here. There is blunt end cut also, I mean straight way cut. Okay, the opposite type is blunt end cut. So sticky end cut is called staggered cut. Anyway, this way sticky ends are produced. Easily it can be joined by the DNA ligase enzyme. So the first step, second step and the third step is joining of this using DNA ligase enzyme. So we have joined both the strands very easily with DNA ligase since the action has produced sticky ends. So this diagram gives you a better idea about how the restriction enzymes act. So here we have discussed the example EcoR1. Okay, EcoR1 is producing a cut in a palindrome with this sequence, and a cut is always in the strand. How it cut is in between G and A on both the strands. Such type of cut is producing what? Staggered end cut is producing sticky ends. This can be very easily stick. Okay, so understand this is the way. First step, cutting action. Second step, the other portion is removed, and from vector and foreign DNA we are with both the sticky ends. In the next session, with the help of DNA ligase, both the DNAs are sticked. Here the sticking will be taking place. Okay, are you okay with this? Easy, right? Okay, so this diagram you have to learn, and this was a board exam question once. With the help of diagram, just show the action of EcoR1. Okay, in the last step we are just showing the DNA ligase enzyme action also. Anyway, keep in mind the action of EcoR1, how it is. Easy, I think. Dear students, in the last class I have given you a textbook exercise to find out more palindromic sequences with examples of enzymes and palindromic sequences. Did you do that? How many of you have worked out that? Your textbook is giving a question, NCERT textbook is giving an exercise question. With the help of your teacher, find out 5 more palindromic DNA sequences, okay, with enzymes name. Okay, so the examples for other palindromic sequences, all of you please note down this to your notebook. Okay, so these are 5 palindromic sequences, examples of 5 palindromic sequences with their enzymes. So you have already studied EcoR1, its palindromic sequence. You know it's GAATTC from 5' to 3' on both sides. It's a palindrome. And the second example is GGATCC. It is for BAMH1. Okay, Bacillus amphifaciens, that you need not expand it, just BAMH1. Then the next example, third example is what? For SMARFIST. Enzyme is SMARFIST. Its palindromic sequence is this one, CCCGGG. Easy to remember. If there are 2 examples, remember this one and EcoR1. It's very easy to remember. CCCGGG. So SMARFIST palindromic sequence is this. It cut the DNA at this particular site only. If there is a sequence like this, the SMARFIST can cut there. It produce blunt and cut, not staggered and cut. Here, hint 3 is the next enzyme. Hint 3, it is cutting the DNA in this particular palindrome. The palindrome is AAGCTT. AAGCTT. Okay. So the last one, 5th one here, the enzyme is HAE, He3. It is cutting the DNA in this particular palindrome. This is the palindrome GGCC, GGCC. Easy to remember. Okay. Anyway, first study the 3 simple one, and then again, since your textbook is giving that question, okay, it may be asked for your board examination. So that is about the restriction endonuclease enzyme. Okay, these are all restriction endonuclease enzyme. Example for restriction endonuclease enzyme: EcoR1, BAMH1, SMARFIST, HINT3, He3. Okay, these are examples of the restriction enzymes, and these sequences are the examples of their recognition site. Okay, where they can produce cut in the DNA. Simple, I think. Note down this and study this. Okay, so the first tool in genetic engineering is a restriction enzyme. Restriction enzymes are of 2 types, exonuclease, endonuclease. For our genetic engineering process, we use endonuclease enzymes which can cut in between. Where they can cut in the DNA, it is specific, and that sequences are known as recognition sites or the cloning sites. These sites are always palindromes, and these are the 5 palindromes. Okay, then the next tool. Before going to next tool, so you just keep in mind, if it's long 2 meters long DNA, our cell is having 2 meters long DNA, we are cutting that DNA. When we use a restriction endonuclease enzyme, wherever this particular sequence is there, all that places this DNA, restriction enzyme cut the DNA. Is it not? If the too long DNA is taken to, Then if we are introducing the enzyme, wherever the palindrome, its palindrome cloning site is there, all it cuts and it produces randomly sized DNA fragments. So keep in mind, understand the action of restriction endonuclease enzymes produces number of fragments, DNA fragments. We cannot say it will be uniformly sized fragment. Okay, where if one palindrome is here, the next palindrome may be distantly placed, and otherwise the next cloning site may be nearly placed. So the action of restriction enzymes produce randomly sized, different sized fragments. Okay, then how we select the fragment we require, the desired DNA fragment, how can we select from this? Okay, that is another great discovery. The process is by the selection. The separation of the fragments is by gel electrophoresis technique. Okay, the technique to separate the fragments. After the action of restriction enzymes, we get number of fragments. How we separate or isolate the desired DNA fragment is by gel electrophoresis technique. We will discuss that technique in our next class. Okay, for today we will wind up. Thank you.